Hi everybody. This lecture is going to cover the biology of personality. As always, I recommend that you have the slides nearby so you can take notes as we proceed through the material. Let's get started. First, I want to discuss basic genetics, which is going to provide some of the information you'll need to understand the information on the future slides. In the cell nucleus of every cell, there are 23 pairs of chromosomes, one from the sperm and the other from the egg. Chromosomes are made up of strands of DNA, and these DNA are grouped into segments that are referred to as genes. Genes, therefore, are the biological building blocks that are passed down. Alleles are one of two genes that are located on a chromosome and that share a specific characteristic, such as hair color. So you can be homozygous for a gene, meaning you have two of the same alleles, or you can be heterozygous for a gene, which means you have one of each. You can see that in the top image on the right. Interestingly, while we're all unique in our own ways, we share the vast majority of our genes with each other, like more than 99%. In the world of psychology, nothing, that I'm aware of anyway, can be reduced to a single gene. That is, there's no extroversion gene. Twins can be monozygotic or dizygotic. Monozygotic twins are commonly referred to as identical twins. They stem from the same fertilized egg, known as a zygote, and develop together in the same placenta. They share all of their genes, so they are often studied to help differentiate between genes and the environment. Dizygotic twins, on the other hand, are basically siblings who developed at the same time. They develop from separate eggs in separate placentas. Ultimately, these twins have the same genetic similarity as regular siblings. They are, however, slightly more similar compared to ordinary siblings because they share certain environmental factors during pregnancy and throughout life. In pregnancy, those factors include the pregnant person's level of stress, activity, diet, etc. Therefore, these dizygotic twins, otherwise known as fraternal twins, tend to be slightly more similar in terms of their personalities than regular siblings, but less so than monozygotic or identical twins. Let's move on to behavioral genetics. Put simply, behavioral genetics is a term used to describe the study of the influence of genetics on behavior. It's essentially the science of gene-environment interactions in terms of their influence on behavior. Heritability is an important concept in the world of behavioral genetics. It is defined as the estimate of genetic influence on a certain trait. It describes the amount of variance on some measure of behavior, temperament, personality, etc. that can be attributed to genetics. The heritability coefficient is a score between 0 and 1 that describes the relative genetic influence of that trait. The higher the number, the more genetically influenced that trait appears to be. The most common approach to studying heritability is through family studies, twin studies, and adoption studies. Family studies look at the presence of traits across family members, such as parent-child, siblings, cousins, aunt, uncles, etc. Heritable traits should have more similarity across first-degree relatives, that is, parents, siblings, and children, than second-degree relatives, like grandparents, aunts, uncles, and cousins. Twin studies usually compare monozygotic to dizygotic twins, which helps to look at the relative influence of genes versus environment. Heritable traits should have stronger correlations between monozygotic than dizygotic twins. If their correlation coefficients are similar, it speaks to the influence of shared environments, which we'll discuss on the next slide. If monozygotic twins do not have a notable correlation in a certain trait, it provides evidence of non-shared environmental factors, which are also explored on the next slide. Adoptive studies compare adoptive children against their adoptive parents and biological parents. If evidence shows that adopted children are more similar to their biological parents than to their adoptive parents in terms of a certain trait, it suggests genetic influence. If the opposite occurs, that is, the adopted child is more similar to their adoptive parent on a given trait, that supports environmental influence. One of the most well-known studies on twins regarding heritability was the Minnesota Twin Study, which measured heritability of personality through the equation H for heredity equals 2 times R of monozygotic twins minus R of dizygotic twins. R stands for correlation. This longitudinal study explored over 1,500 twin families and 350 adoptive and biological sibling families, and found that no matter the situation or the environment, identical twins shared similar personalities, which demonstrated the powerful role of genetics on temperament and personality. The overall heritability of personality was found to be 0.48, which means that nearly half of the variance is explained by genetics. Interestingly, they found that IQ was more heritable at 0.7. This study is not without flaws. For example, the population was not considered representative to the extent needed to truly generalize findings. The findings are also complicated by the fact that adoptive couples tend to be actually quite similar to biological parents in many ways. And it's also very hard to control for the influence of shared versus non-shared environmental influences, which again, we'll look at the next slide. So where does temperament fit into behavioral genetics? That's actually a tricky question. The goal of behavioral genetics is to determine the degree of influence from genes versus the environment. But before they can determine that, they have to decide on what temperament means in the first place. There's been considerable debate about whether there's even a true distinction between temperament and personality, which has become heightened as more and more researchers are appreciating the role of biology in personality. Those who distinguish between the two point to the fact that temperament is more biologically influenced, particularly via genetics, and present at birth. Historically, developmental psychologists have approached the study of temperament as basically the behavioral tendencies of children, while personality was reserved for adults. For example, two big names in the world of temperament, Stella Chess and Alexander Thomas, describe temperament as, quote, primary innate reaction patterns. Some researchers suggest that, as stated by Cloninger et al. 2019, quote, adult personality is essentially a culturally conditioned expression of childhood temperament. Seemingly, the stability of personality can be attributed to temperament, at least to a degree since it's not perfectly stable, whereas the changes in personality over a time appear to be more due to the environment. This aligns with research findings that demonstrate increased consistency in the big 5 personality traits as people age, which might also explain why the term temperament is primarily used for infants and children, whereas personality is used to refer to adult behavioral tendencies and patterns. Ultimately, temperament seems to be the genetic or neurobiological factors that are present at birth and that influence behavior and form the foundation for personality. This speaks to the behavioral genetic contribution of temperament to personality. That is, temperament, even at birth, likely reflects our gene-environment interaction. This is supported by twin study research that has demonstrated more similarity in temperament across monozygotic twins than dizygotic twins. Fisher et al. 2015 reports that approximately 40 to 60% of variance in personality traits is due to differences in temperament. There are many models of temperament, one of which was put forth by Merveld and Asendorf in a book chapter they published in the year 2000. They attempted to combine findings from Thomas and Chess, Buss and Plomin, and Rothbart, who each had pretty popular but separate models of child temperament. They concluded that the common dimensions across these 3 models are emotionality, extroversion, activity, and persistence. Emotionality refers to the tendency to experience emotions and is most similar to neuroticism. Extroversion refers to the degree of sociability versus social inhibition. Activity refers to how active or energetically inclined the child is. And persistence involves attentional and behavioral self-control and self-regulation towards goals and the determination to persist in the face of difficulties. It's associated with conscientiousness. We'll look at another temperament model towards the end of this lecture, which is based on the Fisher et al. Article, and we'll conclude by looking at those research findings which correlate their temperament model with the Big Five. As I mentioned on the previous slide, environmental contribution can be subdivided into shared and non-shared environments. Shared environmental variance speaks to the degree of familial resemblances in temperament or personality that are not attributed to genetics. These might include aspects of family life such as the caregiver's attachment security, methods of soothing, activity level, etc. They include anything that is common to each child, which includes the neighborhood, their socioeconomic status, religious affiliation, pets, etc. It depends on that family, though. For example, there's probably less similarity in children who are 10 years apart, who could have been raised in totally different environments or different socioeconomic circumstances. These common environmental factors are viewed as much less influential than unique non-shared environmental influences. In contrast, non-shared environmental variance speaks to the uniqueness of an individual's experiences in life and explain differences among family members. These might include common aspects of life such as peer groups, teachers, and sports, or notable experiences like a trauma or a car accident or a chronic illness. It also could pertain to birth order and differential treatment by caregivers. I don't know about you, but as the oldest of 4, I definitely noticed changes in my parents' approach to my siblings compared to me. It's not totally cut and dry. For example, a teacher could be a shared environment if siblings have the same teacher in different years. But, as I stated earlier, something that's typically shared, like socioeconomic status, could vary between siblings, especially if there's a large age gap. Sociobiological factors are somewhat common to everyone, as they pertain to evolutionary forces that we tend to share as humans. Cultural factors may vary more, especially if there's a big age gap in which children are exposed to different cultural values. We'll look at these sociobiological and cultural influences on the next 2 slides. Before that, I want to mention that you'll learn more about behavioral genetics and these shared and non-shared environments in this week's Separated at Birth videos based on the Louise Wise Adoption Center twin studies. There are 2 parts to this video, but they are both listed on the same Canvas page, and only Part 2 is required to be viewed. Part 1's there, in case you're interested. Sociobiology refers to the field that explores how behaviors are partly inherited and affected by evolutionary influences like natural selection. It is essentially, like it sounds, the study of social behavior from an evolutionary biological perspective. Another term or discipline that is often used to describe these ideas is evolutionary psychology. One aspect of sociobiology pertains to our motivation as humans, and animals for that matter, to find our right fit. There are clear survival and reproductive benefits associated with living in compatible environments and engaging in compatible behaviors. Not only are we driven to act in certain ways, we tend to gravitate towards environments that suit us and behaviors that help us ensure reproductive success and survival. Selective migration refers to our physical movement to places that suit us, particularly in terms of temperamental factors. Adaptive advantage also speaks to the tendency to act aggressively when feeling threatened, as it represents our historical need to dominate in social settings to ensure survival and rank. Assortative mating is another central concept of sociobiology. It refers to the fact that we tend to first interact with people like us, and then have children with people who are like us. That means, from a heritability perspective, that we increase the likelihood of being surrounded by people with somewhat similar genetics and environments, and then we pass those genes and those environments on. Inclusive fitness is the concept that an individual's purpose is to survive and reproduce, so we engage in behaviors that are advantageous towards those goals. Direct inclusive fitness is what we do to ensure our own reproductive success, such as caregiving for the young so they survive to reproduce themselves. Indirect fitness refers to what we do to help ensure the survival of our species, that is, our gene pool, rather than our individual genes. Some researchers, such as Marsh 2016, have suggested that this helps to explain the personality trait of altruism, which is essentially defined as selfless giving. The image of the prairie dogs is meant to represent this concept. These animals let out a warning call when they encounter a predator, thereby giving away their location and putting themselves at substantial risk of death. They do it because it increases the likelihood that the majority will survive and pass on their genes. On the other end of the spectrum, sociobiology or evolutionary psychology helps us understand the role of jealousy. It was necessary for fending off competitors and ensuring reproductive success. We've discussed that personality is ultimately an interaction between nature and nurture, so let's look at one of the biggest environmental contributors to personality, which is culture. Note that biocultural influences on personality fall under the umbrella of psychological anthropology. This biocultural approach looks at aspects such as gender, weather, climate, nutrition, etc., in terms of its effect on personality. So first, what is culture? Culture refers to a group or society's shared beliefs, customs, traditions, values, etc. We are influenced by what we view in others, for example, what is rewarded and what is punished, and to what degree. We're also influenced by our own individual languages. What words do we have and how do we use them? Even within the United States, there is tremendous cultural variability. Heck, even within California. You might have noticed that on the West Coast, we tend to be more relaxed compared to the Northeast. This also influences behavioral choices. For example, on the West Coast, we tend to dress more casually, think sundresses, compared to the East Coast, think black sweaters. This phenomenon is often referred to as evoked culture, because the hot weather evokes the cultural norm of lighter weight clothing and lighter colored clothing. This is but one example of how cultural norms are developed in response to ecological factors such as climate, and how those norms then affect our personalities and choices. Ecological and environmental factors such as climate and weather affect more than our approach to fashion. Triadnis and Suh, 2002, offer another example and remind us that the degree of cultural influence to some extent depends on the tightness of cultural influence, which is also related to ecology. For example, Singapore has high rates of punishment for violating social norms, and thus much higher rates of conformity to said norms, compared to, for example, the United States, which is a much looser cultural environment where deviations are more tolerated. There are many factors that affect the tightness of a culture, such as the ease of diffusion. For example, it's much harder to diffuse on an island when you're surrounded by water on all sides. Another ecological variable is the mobility of wealth. For example, cattle are easier to move compared to trees. This leads to what Triandis and Suh refer to as a culture of honor. That is, if wealth is movable, we need to be more aggressive to protect our goods from being stolen. Connolly, 2013, provides another example in terms of ecological influence on our basal metabolic rates, which shape personality factors such as assertiveness and aggressiveness. Individuals who live in hotter climates have to acclimate to heat, whereas those in wet, cold, and gloomy climates are more likely to be reclusive. They burn fewer calories because their BMR is less demanding. Ultimately, Triandis and Suh suggest that ecology shapes the culture itself and creates norms for the entire society, which then convert into personality influences. And, as we talked about on the last slide, part of this picture is selective migration, which influences our physical movement towards compatible locations. If all laid-back people are drawn to the laid-back environment of Southern California, it only becomes more laid-back over time. McCrae 2011 acknowledges the role of culture in the expression of the Big Five, particularly in terms of social behaviors, such as health habits, religious affiliations, and vocational interests. With that said, the Big Five has surprising cross-cultural validity. Research such as Renfro 2000 have provided insight into these cross-cultural personality traits. For example, Asian nations were found to be lower in extroversion, South Americans rated higher in openness, and European nations lower in emotional stability. Eon et al. 2017 also explored cross-cultural similarities, but they looked at the hexaco traits. They also found some degree of congruence across cultures. They sampled over 1,600 participants in India, Indonesia, Oman, Romania, and Thailand, all with different languages and religious traditions, which helps to increase cross-cultural variability. Notably, Oman had the lowest congruence at 0.84, while India had the highest congruence at 0.92. Research such as Lee et al. 2000 has also demonstrated some cross-cultural variability in terms of our tendency to be more self-critical or self-enhancing. They found that collectivistic cultures, such as many of the Asian cultures, tend to have higher self-criticism and focus more on the potential negative consequences when thinking about their group membership. In contrast, individualistic cultures, like the US, tend to focus more on the positive aspects of their independence. One example of cultural inconsistency are culture-bound disorders, many of which are beautifully described in Paniagua's 2000 chapter, Culture-Bound Syndromes, Cultural Variations, and Psychopathology. There are numerous examples of culture-bound disorders, so these are just a few examples. Eysenck refers to the American Indian condition in which anger, mutism, and possibly even suicide are prompted by feelings of disappointment or interpersonal conflict. Ataque de nervios is translated to attack of nerves and is associated with Hispanic culture. It refers to the culturally condoned pattern of distress caused by stress and characterized by uncontrollable shouting, crying fits, trembling, radiating heat in the upper body, and verbal or physical aggression. Lastly, Taijin Kyofusho is present in cultural groups throughout Asia. It is characterized by extreme anxiety and/or guilt related to embarrassing others, particularly related to their own appearance, odor, or expressions. As you can imagine, this type of culturally bound disorder makes sense in a collectivistic culture that prioritizes impact on others over self-needs or desires. Hopefully by now you're coming to realize that when it comes to nature and nurture, it's not either or, it's a matter of how they interact together. This is often referred to as gene-environment interaction. While estimates vary, Candler et al. 2019 report that based on nearly 200 behavioral genetic research studies, about 50 to 60% of personality trait variance is attributable to genetics, while the rest is related to environment. Of that percentage that is nurture, they suggest that the majority is due to non-shared environmental influences. Their findings were that at least 40% of variance in hexaco scores is due to genetics, with the smallest genetic components for honesty, humility, and agreeableness, and the highest for openness. But the authors do acknowledge the prominent role of assortative mating in contributing to this finding. They also acknowledge the role of evocation, in that children's behaviors that stem from their heritable temperamental traits likely evoke certain responses from their parents. The similarity of monozygotic twins likely evokes more similar parental reactions, which then makes them seem even more similar genetically than they probably are. The authors refer to this as, quote, reactive nature-nurture, and cite it as a challenge to interpreting twin study findings. On that note, it is important that we realize that there are many reasons why it's difficult to tease apart nature from nurture. Another example is school, which is considered an environmental variable, but the parents decide where to live and where to raise said children, so school is not random. Another part that is tricky is the phenomenon of epigenetics, whereby environmental influences change the expression of genes over time, which we'll discuss next. But first, I want to mention a video that you'll watch this week. Are there personality differences between sexes that bring together genetics and other biological differences with environmental and cultural influences that affect observed differences? So genes are built in, but they are turned on and off in response to experience. A live brain is a changing brain. Epigenetics is the term that refers to these changes in gene expression that occur in response to experience. Your DNA remains the same, but genes are activated or inactivated, which affects which specific proteins are created or not created. You could think of it like a light switch. Certain experiences cause the gene switch to turn on, while others turn it off. Note, these are changes in gene expression, not changes to the genetic code itself. The code for the gene does not change, it's only whether it's activated or deactivated. These changes can be reversible, but they can also be permanent to a degree, and even passed down to future generations. Many environmental factors have epigenetic interactions. For example, stress, nutrition, exercise, sleep, trauma, enrichment opportunities, coping skills, meaningful social interactions, and early nurturing all impact gene expression over time. On that note, you can't spoil a baby. One notable epigenetic trigger is early trauma, which can change the set point for what is known as the HPA axis, one of our primary stress responses. Both Huriana 2020 and Biradelli et al. 2020 Note that epigenetically, early trauma makes the stress response overreactive, which increases the risk of depression, anxiety, suicide, and substance abuse. This is one way in which early life experiences have profound impact on mental health over time and speaks to the importance of emotional enrichment in development. If you're interested in the impact of adverse childhood experiences on future health, I strongly recommend the TED Talk by Dr. Nadine Burke Harris, who is the Surgeon General of California. So, how are epigenetics related to personality? First, through stress, as I just mentioned. It should be fairly clear by now that an overactive stress response would be linked to higher neuroticism, for example. The TED Talk you'll watch this week, The Biology of Our Best and Worst Selves, will provide more insight into some of these epigenetic phenomena that affect behavior and personality. Before moving on, I want to briefly touch on interpersonal or relational neurobiology, which is an interdisciplinary field that explores the intersection of cognitive and affective neurosciences, developmental psychology, and wisdom traditions. It was developed by Dan Siegel, Alan Shore, and Lou Cozolino in the late 1990s. It seeks to understand how relationships in the brain interact to shape the mind and behavior, and emphasizes the role of neuroplasticity, that is, the brain's ability to reorganize itself by forming new connections throughout time. One of the biggest tenets of interpersonal neurobiology is that healthy relationships underlie a healthy brain. It also promotes meditation as a powerful strategy for encouraging positive neuroplastic changes, including strengthening or integrating the connection of the prefrontal cortex and limbic regions that we'll learn about in just a bit. Speaking of the brain, phrenology was the accepted theory of behavioral neuroanatomy until the famous case of Phineas Gage. Phineas Gage was injured at work when an iron rod shot up and penetrated through the left frontal lobe of his brain. While the predominant theory at the time, known as phrenology, was that the brain operates holistically, that is, functions as a whole rather than different parts with different mechanisms, his survival and intact memory, compared to his resulting personality changes, demonstrated the inaccuracy of this view. Therefore, Phineas Gage's accident serves as perhaps the first line of evidence that personality and specific regions of the brain are directly related, and that we can suffer extreme brain damage and remain alive, depending on not only the extent of the damage, but the location as well. His case disproved phrenology's assertion that personality, and intelligence for that matter, were related to the size and shape of the brain rather than the structure and function of it. He was a different person after the accident, the extent to which is widely debated, which speaks to the importance of the frontal lobe in shaping our personalities, intellectual capabilities, and other important functions. On that note, let's take a look at some specific brain regions in terms of their particular functions. Thanks to Phineas Gage and the mountain of research that followed, we know that individual brain regions and coordination of brain regions underlie our personalities and our response to the environment. The first two listed areas are towards the front of your brain, nearest to your forehead, in the frontal lobe, which is one of 4 lobes of your cortex. Let's talk about the prefrontal cortex first, which is at the very front of your frontal lobe. The prefrontal cortex is often nicknamed the executive of the brain because of its role in decision-making, personality, and executive functions, like attention and impulse control. Interestingly, the right and left sides of the prefrontal cortex have very different functions. Whereas the right prefrontal cortex is linked to avoidance and associated with the behavioral inhibition system we'll talk about towards the end of the lecture, the left prefrontal cortex is associated with approach behaviors, an aspect of the behavioral activation system. Another important cortical region is the anterior cingulate cortex, which is located near the middle of the frontal lobes, where the 2 lobes meet. The ACC, as it's commonly referred to, has functions like emotional self-control, social cognition, empathy, reward processing, acute pain perception, decision-making, and impulse control. Therefore, higher brain activity in this region is linked to more focus on so-called, quote, negative emotions like sadness, foreboding, and emotional pain. Not surprisingly then, psychopaths may have low activity here. There's also research that suggests dysfunction implicated in autism. The next 5 areas are limbic regions, which are buried deeper into your brain towards the very middle from all sides. The amygdala is the hub of fear processing and is sometimes referred to as the watchdog of the brain. If you've seen the film Inside Out, it's fairly well represented by the purple character. The hippocampus is important for being present and for encoding new memories. The mnemonic to help you remember this brain region is, if you saw a hippo on campus, you'd remember it. This brain region is particularly susceptible to stress-induced damage and, when damaged, creates problems in terms of creating contextual memories. That means that damage is associated with impaired ability to create context-dependent memories, which leaves us at risk of not being able to recall the specific factors of that situation. This might lead someone who experienced a trauma by a tall male to fear all tall men, rather than to experience fear in response to a more specific person or situation. Briefly, the septum regulates emotional responsiveness and reins it in. Damage to this area can cause septal rage syndrome, which is just as bad as it sounds. The hypothalamus can be thought of as the conductor of the autonomic nervous system and the endocrine system. It tells areas of the body like the pituitary gland when and how to respond to stress. The big neuroscience joke, and there aren't many, is that it regulates the 5 F's: fever, feeding, fighting, falling asleep, and mating. The PC term for mating is fornication. Lastly, for this slide, as there are a number of other limbic regions we won't cover, the nucleus accumbens is an important part of the so-called dopamine reward circuit, which means it is associated with functions like incentivized learning and motivation. The last brain region included on this slide is located in what's called the brain stem, which is, like it sounds, at the base of our brains closest to where it connects to the spinal cord. The brainstem represents the oldest area of the brain from an evolutionary perspective and regulates the most vital bodily functions like heart rate and breathing. Whereas Phineas Gage was able to survive a massive injury to his frontal lobe, he could not have survived the same damage to his brainstem. The noted region here is the reticular activating system, which has a very important role in alertness and directing attention. One of its primary jobs is to activate the cortex when it's time to be alert to something important. So let's briefly discuss the chemicals that regulate or impact the functions in the body, such as neurotransmitters like serotonin, dopamine, and norepinephrine, and hormones like cortisol, testosterone, estrogen, progesterone, and oxytocin. Serotonin is most widely known for its role in mood, largely due to the class of antidepressant medications known as selective serotonin reuptake inhibitors, or SSRIs. While serotonin gets a lot of attention for its mood regulation qualities, mood disorders have been connected to dopamine and norepinephrine as well, along with hormones, actually. Along with mood functions, it also has important roles in regulating impulsivity. Low serotonin activity is associated with higher rates of impulsivity, and particularly impulsive aggression. Higher levels of serotonin are also linked to anxiety, which promotes harm avoidance, a feature of the behavioral inhibition system we'll discuss in a bit. Dopamine is the yang to serotonin's yin in terms of avoidance versus approach. Higher levels of dopamine are linked to the sensation of reward and reinforcement, and thus underlie approach behaviors associated with the behavioral activation system, also discussed later. High dopamine is also linked to increased aggressiveness. While dopamine is released in anticipation and consumption of a reward, it's low dopamine that often prompts us to seek this rush of dopamine as a means of compensation. Therefore, people who have relatively lower dopamine activity are likely to be more impulsive and sensation-seeking. Norepinephrine and epinephrine are 2 chemicals released in response to activation of the sympathetic nervous system, our fight-or-flight response, so to speak. Sympathetic nervous system is the side of the autonomic nervous system that responds to acute, mild stress and causes symptoms such as increased heart rate, sweating, and vigilance. It's our survival response. High norepinephrine is also associated with increased formation of emotional memories and is thus linked to panic attacks and PTSD. This can be contrasted with the HPA axis mentioned earlier, which responds to more severe and chronic stressors and culminates in the release of glucocorticoids like cortisol. Next, we have testosterone, which is the most well-known example of an androgen sex hormone, which can be contrasted against estrogen and progesterone, which are 2 other types of sex hormones. When you watch the Robert Sapolsky TED Talk this week, you'll be exposed to a bit more information about testosterone, and will also be introduced to the MAO gene, which codes for the enzyme that breaks down monoamines such as serotonin, dopamine, and norepinephrine. Testosterone regulates a variety of bodily functions. High testosterone is associated with sensation-seeking, aggressiveness, and antisocial behaviors. While it is fair to say that, on average, biological males have higher levels of testosterone and lower levels of estrogen and progesterone compared to biological females, everyone, with the exception of those with medical conditions, have some degree of all of these hormones. Estrogen also has many functions in the body, one of which pertains to its close relationship with the HPA axis. In fact, high levels of estrogen lead to higher rates of cortisol release, which makes people more sensitive to stress. Estrogen, along with progesterone, also help regulate the metabolism of serotonin, dopamine, and norepinephrine, and they antagonize the neurotransmitter GABA, which is essentially the off switch for anxiety in the brain. Interestingly, less GABA means more dopamine, because it's GABA's job to rein dopamine in. Therefore, individuals with higher estrogen and progesterone compared to testosterone have higher levels of neuroticism, anxiety, depression, and reinforcing responses to substances. Keep in mind that menstruating bodies and those going through pregnancy or menopause are at a heightened risk of mood fluctuation and conditions due to varying hormonal balances. This is one reason why hormonal birth controls often have mood-related side effects as well. Oxytocin is sometimes termed the love hormone due to its connection with so-called maternal behavior and social bonding. It is released during labor and childbirth and during orgasm, and also when we're around people we like and know. It encourages facial processing when we feel safe, and it has an anxiolytic quality most of the time. Whereas testosterone and potentially dopamine encourage more aggressive and competitive stress outlets, estrogen and oxytocin prompt tend and befriend responses, a term coined by Shelley Taylor at UCLA. It essentially refers to the tendency to seek out social support in times of stress. According to Sellers et al. 2007, this appears to be due, at least in part, to the fact that testosterone amplifies the physical stress response and thereby encourages more active, physically related coping, compared to oxytocin, which dampens the physical aspect of the stress response. We're also socialized, as females, to engage in more social bonding activities compared to males. One of my personal biggest pet peeves is when I hear people talk negatively about adolescence. While adolescence gets a bad rap for being the period of development associated with, quote, raging hormones, it unfairly demonizes a stage that is linked to amazing qualities as well. Also, so little of this unique period of development is actually due to hormones. Instead, it's primarily due to changes that take place in the brain and that characterize this stage compared to other periods of the lifespan. Adolescence, which is the period of development ranging from approximately age 13 to 25, is a time of significant biological change. During this period, the brain is undergoing a massive remodeling project that involves two major tasks, pruning and myelination. Pruning refers to removing neural connections that are no longer used and therefore unnecessarily taking up important space and energy. You could think of it as a tree with too many branches. Pruning functions as the clippers that trim the tree's leaves and branches back in order to improve the overall health and functionality of that tree. The other aspect of the remodeling process is known as myelination. Myelin is a fatty substance that coats the tails of many of the cells of the nervous system. This coating helps protect these cells and increases their efficiency when they communicate with others. The more myelinated a certain pathway is, the more efficiently a cell can communicate to another cell, and therefore the more efficiently it can become activated and transmit important information. That's why when we've practiced something a lot, it becomes more natural and easier to accomplish. Miller and Minton 2016 state that pruning and myelination are important aspects of neuroplasticity because they support the brain in reconfiguring itself based on experience. It allows, for example, the frontal lobe to more effectively inhibit impulses that underlie self-discipline. It also helps to explain the use it or lose it phenomenon. If you practice piano a lot from age 5 to 7, but then didn't touch it again, those connections would be pruned during adolescence. However, if you were practicing leading up to, and even better, through adolescence, those connections would have been myelinated, and you'd be more likely to have that skill for the long term. Another aspect of the adolescent brain is the continued development of the prefrontal cortex, which is not completed until around age 25. It's no surprise then, based on this information, that car insurance premiums drop dramatically around this age. They know your brain is a safer driver. As we talked about a couple of slides ago, the prefrontal cortex's job, among others, is to regulate the limbic regions. Therefore, one aspect of adolescence is higher rates of impulsivity, moodiness, and emotional reactivity. On a positive note, this also contributes to the unique sense of passion, social engagement, and creativity that characterizes this developmental period. Another feature of the adolescent brain that contributes to this passionate exploration is the low baseline level of dopamine and the high dopamine response to reward. Adolescence is, after all, the time when we leave the so-called nest, and as such is associated with seeking novelty and trying new things. On the positive side, this exploration is exciting and we feel energized and alive by it, which helps to compensate for the more intense susceptibility to boredom caused by the low baseline dopamine activity. On the other hand, adolescence is a time where they're more likely to heavily weigh the positive benefits and to disregard or minimize the potential risks of a decision, which is referred to as hyper-rational thinking. This makes adolescence a period of significant risk-taking and sensation-seeking, which is further amplified by the big dopamine rush they get from moving towards these rewarding activities. It also promotes activities like substance use as a strategy for achieving the dopamine rush they're actively seeking. If you're interested in this topic, I highly recommend Dan Siegel's book, Brainstorm, that discusses all of this information in more detail and provides suggestions for how to best support adolescents at this time. I've mentioned stress a few times in this lecture so far, which speaks to the powerful influence that stress has on personality. I am fascinated by the role of stress and focus on it quite a bit in my biopsychology courses. So if you're interested in learning more about the biopsychology of stress, my lecture videos are available on my YouTube channel. First, let me say that it's stressful to try and live a stress-free life. Stress is a core feature of being alive, and we can't escape it. But we can learn how to maximize our approaches to healthy coping. Keep in mind that stress not only affects us in the moments we experience it, but also more generally via epigenetic mechanisms. Stress, both directly and indirectly, affect the monoamine system, that is, serotonin, dopamine, and norepinephrine. This is one of the reasons why stress is linked to mood volatility, that is, mood swings. Stress can occur in response to psychological triggers, like feeling judged, but also to physiological ones, like being tired or hungry or cold. If you've heard the term hangry, you know about this connection already. Note that this is one of the many explanations for situational specificity. You might be an otherwise very agreeable individual, but still snap at someone when you're super hungry and tired. Also note that stress encourages defensiveness and fighting by decreasing the prefrontal control over limbic regions. It is okay to sleep on it. I mentioned a bit ago that high dopamine and high estrogen both increase the risk of substance use disorders, colloquially known as addiction. First, stress increases this risk by both promoting our desire for escape from unpleasant feelings, thoughts, and sensations, but then secondly, by increasing the rewarding nature of the substance and other rewarding activities for that matter, like gambling, sex, and shopping. This occurs via brain oxygenation, part of the stress response, that leaves us feeling more alert and alive. And we also get a big rush of dopamine that feels particularly rewarding and reinforcing. Substances like alcohol and Xanax further complicate this picture because GABA, the off-switch neurotransmitter, alleviates our sense of anxiety, which teaches the brain to seek out substances as an escape mechanism in the future. The use of substances to avoid or escape something unpleasant is an example of negative reinforcement, which is known to be riskier in terms of addiction than positive reinforcement, which refers to the motivation to gain something pleasant. It's essentially the difference between drinking to avoid anxiety compared to drinking for fun with friends. Keep in mind that substances could easily be replaced by workaholism, sleeping too much, distraction. There's lots of ways to avoid. Fun fact, stress also enhances memory formation, which makes it more likely that we'll remember the seemingly positive feelings of these substances or whatever else we're escaping with. And of trauma for that matter, which is why stress-induced hippocampal damage that impairs our contextual memory formation is so problematic. We don't want to generalize our triggers. I'll touch on that again at the end of this slide. Another aspect of stress that affects our personality is sleep. Stress makes it hard to sleep because the stress response is stimulating. It's meant to promote survival by running or fighting. Then, when we're tired, it's a source of stress in and of itself. So a vicious cycle is easy to create. You're stressed, so you can't sleep. So you're tired, so you're stressed, so you can't sleep. It also tends to be the case that people who have trouble sleeping will begin to stress about their ability to get enough sleep, which only further complicates this picture. And personality-wise, being tired is a major physiological trigger of mood volatility, irritability, and impulsivity. As I've mentioned, stress can cause sympathetic nervous system activation, the adrenaline rush of norepinephrine and epinephrine. But in response to more chronic and or severe stressors, the HPA axis takes over. High activity of the HPA axis leads to high levels of cortisol release that is associated with higher neuroticism for a variety of reasons. First, chronic activation of the amygdala watchdog makes it bigger and stronger over time, which means it barks at more things, and it's louder and it barks longer. Next, hippocampal damage impairs memory formation, which leads to more general memories, which means more generalized triggers. It also means that the HPA axis has trouble turning itself off because the receptors are located in the hippocampus. Similarly, the prefrontal cortex undergoes volume loss, which makes it harder to regulate limbic regions by inhibiting the primitive responses of these limbic areas. It also impairs fear extinction, which refers to the ability to learn that something doesn't need to be feared. The damaged prefrontal cortex and hippocampus together also have important roles in evaluating whether the stress response is even warranted. So volume loss in these areas results in longer sympathetic activation as well. On a positive note, these changes are not necessarily permanent, and there's a lot we can do to compensate and change it for the better, including therapy and exercise. I know this is already a very long lecture video, but I also know from experience that many students feel insecure about biological material, and thus I wanted to provide more detail than I might normally. If you do have a strong background in biology, you might be able to watch this on double speed. So, before we finish up this lecture, I want to briefly talk about some of the more prominent theories of personality from a biological perspective, starting with Sheldon's theory of somatotypes. William Sheldon began studying the relationship between the structure of the body and an individual's temperament in the 1940s and 50s, based on the earlier work by Ernst Kretschmer in the 1930s. Sheldon developed 3 body types that tend to correlate with 3 personality types. Note that Sheldon believed we all have parts of each. It's the combinations of degrees to which we fit each body type that determine our stereotypic temperament. He acknowledged that genes, in addition to environmental factors like nutrition, affect our somatotypes. Ectomorphs are described as anxious, self-conscious, emotionally sensitive, quiet, artistic, thoughtful, and appreciative of intellectual stimulation, much more so than social interactions. Sheldon proposed that individuals during embryonic development have more ectoderm development, which is the outer layer that develops into skin and the nervous system, hence cerebrotonia. Cerebrotonia is essentially a pattern of temperament that is drawn more towards intellectual stimulation than social or physical stimulation. Mesomorphs are most similar to the type T sensation seekers we'll be discussing next. They are curious, assertive, agile, competitive, alert, and can be fearless and aggressive. Mesomorphs, considering their body types, likely have higher levels of testosterone that influence their potential aggressiveness, which could be channeled into pro-social, non-problematic activities like extreme sports. Sheldon theorized that these individuals during prenatal development have stronger mesoderms, or middle layers, that become muscle, hence somatotonia. Somatotonia is considered the pattern of temperament in which there's a preference for physical stimulation over intellectual or social stimulation. Lastly, endomorphs, who tend to have narrow shoulders compared to their wider hips and carry more fat on their bodies and their hips and gluteal areas, similar to a pear shape, compared to ectomorphs and mesomorphs. These individuals tend to be more easygoing, social, relaxed, affectionate, and resistant to disapproval. They want to enjoy life, and they love food and other more hedonistic pleasures. During prenatal development, Sheldon suggested that they have stronger inner layers, or endoderms, which develop into viscera. Visceratonia, therefore, is essentially viewed as a temperamental pattern of preference for social stimulation over intellectual or physical stimulation. As I stated earlier, there can also be combinations. So somebody might be described, for example, as an ectoendomorph or an endoectomorph. Interestingly, Eysenck, who we'll discuss shortly, was unable to prove his findings, but other studies have provided some support for Sheldon's somatotypes. So the jury's still out there as to how accurate this theory may be, and it's likely that there are some correlates, but certainly not a perfect theory. Type A and B personality types were coined by the cardiologists Meyer Friedman and Ray Rosenman in the 1950s. Type A was noted as a potential risk factor for heart conditions compared to Type B. Type A's are described as competitive, self-critical, easily agitated, and having a strong sense of time urgency. These individuals strive towards goals without feeling a sense of joy in their efforts or accomplishments. They pose a danger to themselves because of their negative view of themselves and tendency to be easily aroused to anger or hostility. Type A's have a tendency towards repression and hostility. Repression is consistent with hyperfrontality, that is, an overactive frontal cortex, which leads to a tight inhibitory grip on the limbic regions and is consistent with Type A's preference towards structure, rules, planning, discipline, and black and white thinking. Repression is also linked to an overactive stress response, which is consistent with the cardiac risk that characterizes this type. Hostility is also linked to poor health outcomes. It appears to stem from a sense of time-pressuredness and repressed anger. It also appears to encourage poor lifestyle choices and limited social support, as people tend to avoid those who come across as hostile, and therefore provide less support to them. Type B personalities, on the other hand, are described as relaxed, patient, and easygoing. They are more tolerant of those around them and tend to be well-liked and empathetic. Type T personalities, which was not one of the original types, was added by Frank Farley. It is sometimes referred to as Big T, T for thrill-seeking, but you could also think of it as T for testosterone, because it is linked to higher levels of this hormone. It's also related to dopamine. These individuals tend to be thrill-seekers and thrive on variety, complexity, intensity, and conflict. Farley differentiated between the positive side, such as engagement in extreme sports and entrepreneurship, and the negative side, such as engagement in crime, violence, and overuse of substances. We'll explore more about risk-taking as it pertains to sensation-seeking on the next slide. I've referred to sensation-seeking multiple times throughout this lecture, so let's skydive into that, pun intended. Sensation-seeking is, just like it sounds, the trait associated with a strong drive towards living life to the fullest through novel and exciting experiences. Individuals who are high in this trait tend to be risk-takers who resist the pressure to conform to social norms and who have limited inhibitions. It's a central characteristic of the Type T personality. Farley argued that Type Ts are more prevalent in the U.S. because of our culture of immigrants and how it colors our society. It takes a lot of courage to face the risk of immigrating to a new country. From a biological perspective, it is related to high testosterone and low dopamine. Genetically, some of the predisposition has been linked to the D4 dopamine receptor gene, which codes for the proteins that dopamine binds to and affects how sensitive those receptors are to dopamine. Interestingly, ADHD is also linked to differences in the D4 dopamine receptor gene, which underlies both its heritability as well as the typical impulsivity and risk-taking characteristics of untreated ADHD. Physiologically, low dopamine prompts sensation-seeking as a mechanism of compensation, since the excitement of seeking novelty and the consumption of exciting experiences result in an additional dopamine rush. According to Holmes et al. 2016, there may also be a link between sensation-seeking, motor impulsivity, and reduced cortical thickness, specifically localized to regions implicated in cognitive control, like the anterior cingulate cortex and the prefrontal regions known for mood regulation and emotional expression. From a lifespan perspective, sensation-seeking increases during childhood, peaks at adolescence, and declines throughout adulthood. That should make sense in light of the unique biological aspects of adolescence discussed a few slides back. Sensation-seeking is considered to be both a stable trait as well as a changeable one. We probably all know someone who can be easily characterized as high sensation-seeking. That family member or friend, or maybe it's you, that dreams of hiking Everest, skiing off of a helicopter, or swimming with sharks. This speaks to the stability of the trait, that overall, some people just typically crave these experiences more than others. In contrast, sensation-seeking can represent a variable trait. This is more likely to occur in someone who is moderate in the overall trait, which makes them more influenced by situational factors. Perhaps when on vacation and life is good, they're open to the occasional sensation-driven experience, but for the most part, they don't engage in these behaviors and they're fine with that too. Arguably the most well-known measure of sensation-seeking is the Zuckerman-Kuhlmann Personality Questionnaire. It measures what the creators refer to as the, quote, alternate five-factor model of personality, which includes impulsive sensation seeking, sociability, neuroticism, anxiety, aggression, hostility, and activity as the 5 dimensions. The impulsive sensation seeking trait has 4 components: experience seeking, thrill and adventure seeking, disinhibition or impulsivity, and boredom susceptibility. If you're interested, this scale is provided in your module as an optional assessment this week. Hans Eysenck was mentioned in the trait theory lecture as an example of a researcher who used factor analysis to determine dimensions of personality. His theory was saved for this lecture because he was very vocal about the primary role of biology in influencing personality. Consistent with his emphasis on biological contributions, Eysenck focused on temperament rather than personality. He believed that personality stems primarily from genetics and physiology, which determines our inborn temperament. His model, which is commonly referred to as the Penn model, or sometimes the 3-factor model, includes 3 superfactors, as he called it. Notably, his original theory only included the E and the N. P was added in conjunction with his wife Sybil Eysenck later on. The original theory created 4 quadrants by intersecting 2 dimensions. Going right to left is extroversion to introversion, and going up to down is neuroticism to emotional stability. As you can see in the chart, this creates 4 quadrants, which include a variety of traits in each. These 4 quadrants also correlate with the ancient Greek theory of the 4 humors, which is popularly associated with Hippocrates. Their belief was that the strength of various fluids in the body, known as humors, directly affect an individual's temperament and health. The 4 types correlate with these humors and are based on the intersection of temperature and humidity. Moving clockwise from top right, you'll see choleric, which is high yellow bile, and warm and dry. These people are described as having a hot temper, aggressive, tense muscles, and yellowish. Choleric temperaments are angry, short-tempered, irritable, and this is due, presumably, to an excess of yellow bile. Sanguine personalities have high blood levels and are described from the intersection of warm and wet. They are cheerful, optimistic, pleasant, comfortable, and have rosy cheeks and a healthy glow. Sanguine temperaments are ultimately cheerful and optimistic, which again is due to an excess of blood. Phlegmatic personalities have high phlegm or high mucus and are cold and wet. Cold, being kind of slow, lazy, and dull. Phlegmatic temperatures are calm, relaxed, and peaceful, which is apparently due to having an excess of phlegm. Melancholic personalities have high black bile. While there is no such thing according to modern medicine, it is unclear what they were actually referring to. These personalities are cool and dry and are described as sad and pessimistic. They're depressive, analytic, and quiet. And again, this is apparently due to an excess of black bile. Okay, let's get back to Eysenck. As I mentioned earlier, in the top-down hierarchical Penn model, there are 3 superfactors, which are considered to be the most stable aspects of personality. They include psychoticism, extroversion, and neuroticism. Below that are traits, which correlate with individual superfactors and are slightly less stable than the superfactors above it. Levels and combinations of these temperaments lead to different levels of specific traits, such as how talkative, thoughtful, pessimistic, or impulsive we might be. Note again that while temperaments are considered to be more biologically influenced, specific personality traits are largely affected by environmental factors. These include the terms in each box, such as aggressive, sociable, passive, and moody. Below traits are habits or recurring behaviors, which are more transient than traits, and at the very bottom of the hierarchy are single occasion behaviors, which are the most prone to situationalism. Let's go through each dimension separately. The extroversion-introversion spectrum is differentiated between excitation and inhibition. The extroversion dimension reflects differences in cortical arousal due to differences in activities of the ascending reticular activating system, ARAS. As a reminder, it's the reticular activating system's job to activate the cortex. Therefore, high ARAS activity results in higher cortical arousal. In fact, this has sometimes been referred to as an arousal or activation theory due to the emphasis on biological stimulation. Unlike extroverts, introverts have higher baseline cortical arousal due to higher ARAS activity. This leads extroverts to crave excitement. They get bored easily. They are naturally under-aroused, so they seek out opportunities to restore that balance via sensation seeking, compared to introverts who try to restore balance by controlling their surroundings and quieting things down. Introverts are overall more alert and less able to check out. Eysenck described them as, quote, chronically aroused compared to extroverts. While extroverts are social, outgoing, and readily connect with others, introverts have a greater need to be alone, engage in solitary behaviors, and limit their interactions with others. To some extent, this dimension corresponds with what's known as the Yerkes-Dodson law, which proposes that we're at our best at a moderate level of arousal. Too much arousal and we feel overwhelmed. Too little, we're unmotivated and careless. What we need as individuals to achieve moderation differs. We might need stimulation, or we might need the opposite. The neuroticism stability spectrum ranges from calm people on one end to highly moody and emotionally reactive on the other. Eysenck explained these differences in terms of sensitivity of the sympathetic nervous system and response to sympathetic activation. Neuroticism is, therefore, associated with a more easily triggered limbic system, which results in more frequent and more intense sympathetic activity. It is presumed that someone high on this dimension has an activation threshold that is lower than somebody who is more emotionally stable. Essentially, Eysenck theorized that people who score higher in neuroticism have more active fight-or-flight responses. This helps to explain why someone like me feels tortured by a horror movie compared to someone else who has a much less active sympathetic nervous system and craves that stimulation. It's important to note that introverts and extroverts can be very different depending on their level of emotional stability, since one trait modifies the expression of the other. It's also important to note that high limbic activity stimulates ARAS, which stimulates the cortex, so there's reason to believe that neuroticism and introversion may be related in some ways. Psychoticism was added last in 1976 while working with his wife, Sybil. First, I want to say that high scores on this scale does not mean someone is psychotic, just that they are more prone to it and or share features with it. Just like a high score in neuroticism doesn't automatically mean you have an anxiety disorder. High psychoticism is associated with having less empathy, more aggression, less social conformity, more hostility, more impulsivity, and more troublemaking behaviors, all of which are associated with high testosterone. They tend to be independent thinkers who are less responsive to conditioning, and again, therefore less likely to adapt to social norms. This makes them more likely to engage in illegal activities, but also more likely to be highly creative. On the other end of the spectrum, which is sometimes referred to as normality, or impulse control, or socialization, there is an association with altruism, empathy, cooperation, impulse control, and conventionalism. Psychoticism is negatively correlated with honesty, humility, agreeableness, and conscientiousness. Hans and Sybil worked together to develop a measure of their Penn model, which they named the Eysenck Personality Questionnaire. Jeffrey Gray is one of the most well-known neurobiological personality theorists. He was a student and successor of Eysenck's. Not surprisingly, then, his view of personality was also heavily grounded in the biological foundation of temperament. His theory, known as reinforcement sensitivity theory, has been revised multiple times, with the most recent revision, to my knowledge, by Gray and McNaughton in 2000. Gray noted that people who are high in impulsivity tend to be sensitive to reward compared to those who are high in anxiety and tend to be more sensitive to punishment. His research led to the reformulation of Eysenck's theory to focus on three bodily circuits: the behavioral activation system, behavioral inhibition system, and fight-or-flight system. Unlike Eysenck's top-down hierarchical approach, Gray viewed these three systems from a bottom-up perspective, that these three systems serve as the foundation from which personality is built. The behavioral activation system involves activating limbic circuits in response to conditioned signals of reward, which activates dopamine in order to facilitate approach behaviors. More recent research implicates endogenous opioids like endorphins, essential to this circuit also, but we'll leave it as dopamine for the purposes of this course. The behavioral activation system, therefore, is sensitive to appetitive experiences related to something viewed as desirable or rewarding. It's associated with the feeling of hope, excitement, and joy, and it's action-oriented. Individuals who have highly sensitive BAS reactivity tend to be more extroverted and open to experience. Keep in mind that this system is activated in response to something incentivizing. They want to move towards rewards and will put substantial time and energy into doing so, often spontaneously, but also sometimes with planning. These individuals are prone to sensation-seeking, risky behaviors, and impulsivity, which increases the risk for addictive behaviors and patterns. Impulsivity, in particular, appears to be a function of reward sensitivity and is correlated with extroversion and slightly with neuroticism. They tend to be more willing to experience pain and to view pain as part of their anticipation of reward rather than as an indication of danger. The behavioral inhibition system is very much the opposite of the activation system. Stronger BIS reactivity is associated with anxiousness, avoidance, and a higher perceived sensitivity to threat, to conditioned punishment. That is, what we've learned to be unpleasant. These individuals are very sensitive to potentially aversive experiences and will immediately inhibit ongoing behavior, arouse the cortex, and direct it to analyze the situation. While the BAS is related to dopamine activity, the BIS is correlated with serotonin. Correspondingly, BAS activity drives approach behaviors and fixation with positive outcomes, while the BIS drives avoidance and preoccupation with negative outcomes. Individuals high in BIS sensitivity, therefore, are quite more prone to anxiety, and particularly more to generalized and social forms of anxiety that have strong cognitive components. Moreover, not surprisingly, BIS activity is correlated with higher neuroticism and more fear of experiencing pain due to the interpretation of pain being a sign of danger and therefore warranting avoidance. It's also slightly more correlated with introversion compared to the BAS's relationship with extroversion. Lastly, the fight-or-flight system is a more fear-based version of the BIS. Whereas the BIS responds to learned or conditioned threats of punishment, that is, anxiety, the fight-or-flight system is a physical response to unconditioned aversive stimuli, such as a threat to life, which reflects fear. This might be the difference between BIS reactivity to a partner saying, "we need to talk," versus FFS responding to seeing a bear while hiking. Note that while Gray differentiates them on this basis, there is controversy as to whether the BIS and FFS can actually distinguish between conditioned and unconditioned stimuli. FFS activation is akin to the sympathetic nervous system being activated as a survival-oriented response, including increased heart rate, respiration, muscle tension, and GI distress. It triggers us to be more aggressive or defensive rather than avoidant. The research shows that some people are more inclined to respond with freezing or fainting, which is outside of the scope of this lecture, but worth mentioning. This aligns with Eysenck's neuroticism in that people with high FFS activity tend to have lower thresholds and more extreme sympathetic responses. It also appears to be related to Eysenck's psychoticism dimension in terms of the evoked potential for aggressiveness. We'll finish off this lecture by briefly looking at the relationship between temperament and the Big Five. Helen Fisher at the Kinsey Institute is a well-known biological anthropologist who has contributed to our understanding of the biology of personality and temperament. To borrow a quote stated by Rothbart et al. 2000, stated in her Fisher et al. 2015 article, temperament arises from our genetic endowment. It influences and is influenced by the experience of the individual. One of the outcomes is the adult personality. As a biological anthropologist, she set out to develop a measure of temperament rooted in biology that helps to explain personality. The Fisher Temperament Inventory, according to Fisher et al. 2015, is the first measure of temperament that was constructed based entirely on brain science using functional MRI research and our good friend factor analysis. It accounts for 4 major neuromodulating systems we've discussed in this lecture: dopamine, serotonin, testosterone, and estrogen and oxytocin. Their research led to 4 temperament scales: curious energetic, which is related to dopamine, cautious social norm compliant, related to serotonin, analytic tough-minded, correlated with testosterone, and prosocial empathetic, linked to estrogen and oxytocin. The numbers on the slide beneath each scale represent the correlation between that temperament scale and the Big Five trait. I've only listed the statistically significant results. Notably, recent research by Barrell et al. 2020 have demonstrated that adult attachment security moderates the relationship between temperament and the Big Five, something you'll learn more about in the Object Relations and Attachment unit of this course. Since you'll be reading the Fisher et al. Article this week, I won't go over the findings here, but they're noted on the slide so that you have a quick reference. And, of course, if you have any questions, you know where to find me.